I wanted to talk about maybe some broader implications of the widespread use of glyphosate. You mentioned in the book that these researchers were looking at detecting glyphosate in a Petrie dish, and they had the control, and then they had the glyphosate, the one that they were going to test, and they found glyphosate in both. And you mentioned that the glyphosate is actually in the agar, the substrate that they use, because they're using, potentially glyphosate contaminated substrates in the agar. So many studies use plates like that and potentially have been contaminated with glyphosate. What do you think the broader implications of having so many studies not taking the glyphosate contamination into account?
I think it's going on too with studies on fats. They try these different things where they feed different kinds of fats. They talk about saturated fat versus unsaturated, vegetable oil, and they'll do these experiments with people and try to judge, the quality of these different fats. And unless they look at how much glyphosate is in the fat, they're going to get an answer they don't understand, because it really, I think the critical thing is how much glyphosate is in the fat is what's going to make a huge difference in whether that's a healthy fat or not. It's true for many experiments that are going on. I think people get contradictory results because someone does a rat study and they just say in these studies, they talk about a high fat diet. They give the rat a high fat diet. Often the paper doesn't say anything more than that, and I'm like, well, gosh, I don't know where did they get that fat? Where's it from? Were they using glyphosate? I ask all these questions, and it's not there in the article because they think it's irrelevant. They're not even thinking about it. I think that's happening in lots of studies. And of course, there was that one that was supposedly proved. I don't know if you remember me writing about that in the book, the one that supposedly proved that glyphosate doesn't substitute for glycine. Was that the one you were talking about?
That's the one I was referring to.
Yeah, because that was so interesting because they started out with these cells, which were breast cancer cells, and they had been maintained in culture for decades. So they were very old cells. And while they were maintained, they were probably being fed glyphosate all the while. So they probably had all kinds of glyphosate embedded all over their proteins. I suspect that both the treated group and the untreated control already had lots of glyphosate in their so-called tissues, right, in their proteins. And then when you grow them, they are going to break down the proteins that they have and make new proteins out of them. And they're going to free up glyphosate and put it into the new protein. So both sets of cells were doing that. And then meanwhile, they exposed one set to glyphosate, but there are many conditions in which you can expose cells to glyphosate and it doesn't go in. You can make the conditions such that glyphosate doesn't get taken up by the cells. Whether they did that or not, I'm not sure, but it was certainly possible that the extra glyphosate they exposed them to did not get taken up, but the cells already had plenty of glyphosate inside them, stuck inside proteins from their many years of being exposed to it. So it turned out both sets of cells were able to make proteins that showed up to have their method was really fun. And I was like excited about it because I think someone else could do that experiment if they were a chemist, do it with a better control, to see, because they found a number of proteins and beautifully they had the figure where they actually labeled the sequence. So you could look up the sequence and find out what the protein was, which I did. And I have in my appendix of my book, I actually list all these proteins. And they turn out to have, many of them have what I call the glyphosate susceptibility motif, so they're actually very nicely supporting my notions of where the glyphosate would show up. They found specific places and specific sequences in specific proteins that they reported on in that. But both sides, there wasn't a noticeable difference between the treated group and the untreated group with respect to the amount, which was why they said, well, all of it was noise, all of it was bogus, was what they concluded. But I concluded, well, both of them started out with the glyphosate and they just put it into those proteins. So we don't yet know who's right, but it's very interesting.